Welcome students. In last class we have completed the disaccharides and polysaccharides, their structures and the examples. Today we will see the importance of carbohydrates. These carbohydrates are the major source of energy and a fuel. They are found in animals as well as plants, in human beings also, in the form of storage saccharides. Cell wall of bacteria and plants is made up of cellulose. Even they are found in nucleic acids as D-ribose and 2-deoxy-D-ribose, that is in DNA and RNA. So carbohydrates are formed in biosystems in combination with many proteins and lipids. Now we are going to learn another biomolecule, that is proteins. Proteins are the most abundant biomolecules in the living system. The sources are milk, cheese, pulses, peanuts, fish, etc. It is generated from the word proteose, that is Greek word. Proteose means primary or prime importance. It occurs in every part of the body and form the fundamentals of basis of a structure and functions of life. Also required for the growth and maintenance of the body. So proteins are very essential for our body structure. All proteins are the polymers of alpha amino acids, means the basic structure of proteins is alpha amino acid. So let us start with the study of amino acids. Amino acids, the word itself suggests. Amino means NH2 group and acids means carboxylic acid group, that is COOH group. So the compound containing both amino group and carboxylic acid is called as amino acids. Here the structure is given. The carbon atom is linked to COOH group and NH2 group. One of the valence is satisfied by hydrogen and one of the valence is satisfied by R. So R may be H, it may be CH3, it may be C2H5, or it may be a long chain of carbon atoms, or even it may be aryl group, that is aromatic group. If both the groups are attached to the alpha carbon atom, they are called as alpha amino acids. Now what is alpha carbon atom? The carboxylic acid contains carbon atom, that is numbered 1, and beside that, next to that is called as alpha carbon atom. So here both the groups, that is COOH group and NH2 group, are attached to the alpha carbon atom. Hence they are called as alpha amino acids. There are 20 naturally occurring amino acids. If you want the detailed structures or names, you can refer the table number 14.2 on page number 412. How the amino acids are classified? Based on the chemical nature, amino acids are classified as acidic, basic and neutral. As we have already seen, in amino acids both the groups are present. If carboxylic acid groups are more compared to amino group, then it is called as acidic amino acid. Example, aspartic acid or glutamic acid. If amino groups are more than carboxylic acid, then it is called as basic amino acid. Example, lysine, histidine, etc. But if both the carboxylic acid and amino groups are equal in number, then it is called as neutral amino acids. Example, alanine or glycine, etc. The 2-mark question can be asked as define acidic amino acids and give an example, or One mark question can be asked as give an example of either acidic, basic or neutral amino acid, etc. One more classification, that is based on dietary requirement. As we have already seen, proteins are essential, or proteins forms the basic structure of our body, and these proteins are made up of amino acids. So some of the amino acids are synthesized by our body itself, or some of the amino acids we have to get it from our food or the diet. So based on that dietary requirement, they are classified in two types, that is essential amino acids and non-essential amino acids. The amino acids which are not synthesized in the body and must be obtained through diet are called as essential amino acids. Example, lysine, histidine or phenylalanine, etc. And non-essential amino acids are synthesized in the body. Example, alanine, lysine, aspartic acid. Now here the question can be asked for 2 marks. Define essential or non-essential and give an example, or classify the given amino acids in essential and non-essential amino acids. Let us see the properties of amino acids. Amino acids are colorless, crystalline solids. They are water soluble and having high melting point. They behave like salt rather than simple amines or simple carboxylic acids. Due to presence of both acidic and basic groups in the same molecule, means they are showing both the characteristic as acids as well as bases. When they are dissolved in water, that is in aqueous solution, the carboxyl group can lose a proton, that is it gives H ion, and amino group accepts a proton, giving rise to a dipolar ion. Di means two, polar means charges. So here one is positive and another becomes negative. So such type of dipolar ion is known as Zwitter ion. This Zwitter ion is neutral but contains both positive and negative charges. Now let us see the structure of Zwitter ion in next slide. Now this question can be asked, what is Zwitter ion? So you can write this definition, or one more definition is given. The dipolar ion of amino acid which neither moves towards anode nor towards cathode under the influence of electric field is called a Zwitter ion. Now because of presence of both negative and positive charges, it neither goes towards anode, that is positively charged electrode, or cathode, that is negatively charged electrode when the electric field is applied, that is battery is applied. So such compound or such ion is called as Zwitter ion. Now here the structures are given. Amino acid structure is given as well as Zwitter ion structure is given. Just go through this Zwitter ion structure. You can see COO, O becomes negatively charged, that is COO complete group becomes negatively charged, and NH2 adds a proton or accepts a proton and becomes H3N plus. You can see here it is NH3 plus, and this COO becomes minus. So this is how the positive charge as well as negative charge both are present in the same compound. In Zwitter ionic form, amino acids show amphoteric behavior, as they react both with acids and bases. Except glycine, all other naturally occurring alpha amino acids are optically active, means they rotate the plane polarized light. Since alpha carbon atom is asymmetric, we have already seen which is alpha carbon atom. So this is asymmetric, means chiral, that is the carbon atom which is attached to 4 different groups or atoms. See here, this is alpha carbon atom. One valence is satisfied by COOH, one valence is satisfied by NH2, that is amino, one valence is satisfied by H, and another fourth one is satisfied by R. Now here, if R is different from all these three, then it is then this carbon atom, the middle carbon atom, acts as a symmetric carbon atom or chiral carbon atom, and it rotates the plane polarized light, that is it shows the optical activity. Now how these are existing? These exist both in D and L forms. Most naturally occurring amino acids have L configuration. Now what is L configuration? Is NH2 group is on left hand side of the alpha carbon atom, that is called as L configuration. So here both the forms are given, D amino acid and L amino acid. The question can be asked on Zwitter ion part. What is Zwitter ion? Or give the structure of Zwitter ion. Remember, in Zwitter ion you should show both the charges, that is negative charge as well as positive charge, and when you are showing positive charge on nitrogen, it should be NH3, not NH2. As we have seen that proteins are made up of amino acids. So they are also called as polymers of alpha amino acids. And how these are connected? Polymers means having long chain. Poly means many, mer means chain. So when they are forming the chain, they are connected by the bond, and that bond is called as peptide bond or peptide linkage. Now here the question can be asked for 1 mark. What is define peptide bond or peptide linkage? The amide linkage, that is CONH linkage between two amino acids, is called peptide bond or peptide linkage. The reaction proceeds with loss of water molecule and product is known as dipeptide. If two amino acids are combining, then the product is named as dipeptide. If 3 amino acids are combining, then the product name is as tripeptide. If more than 10 amino acids are combining, then it is called as polypeptide. So one of the example is given in here, that is glycine plus alanine. Now in this structure you can see, in glycine R group is replaced by H, and in alanine R group is replaced by CH3. So OH from glycine combines with H from alanine, loses water molecule, and the remaining structure get joined to each other. This is how the product is formed, which is dipeptide, that is glycyl alanine, and the bond between this, that is CONH linkage, this is called as peptide bond. The product is dipeptide. Now this can be written as even gly dash ALA, that is alanine. So first 3 letters of the amino acids. This is how A peptide is formed by combining more than 10 molecules of amino acid is called polypeptide. So generally we can give a sentence as if n number of amino acids are combined, n minus 1 peptide bonds are formed. Now why I have given this sentence? Because the question can be asked, if 4 amino acids are combining, then how many bonds are formed? So the answer should be 3. A polypeptide containing more than 100 amino acid residues means such 100 amino acids when they are combining and having the molecular mass higher than 10,000 unit is called a protein. So this is the definition of protein. All proteins are polypeptides, but all polypeptides are not proteins. Example, insulin containing 51 amino acid is a protein which is present in our body and it maintains the blood sugar level. So how these proteins are having the structure? Broadly, the protein structure can be given as primary, secondary, tertiary and quaternary. In primary structure, how the amino acids are linked, which amino acids are linked, that idea can be given as primary structure. Proteins may have one or more polypeptide chains. Each polypeptide in a protein has amino acids linked with each other in a specific sequence. This word is very important. Specific sequence of amino acids, that is said to be primary structure of that protein. Any change in this primary structure, that is sequence of amino acids, creates a different protein. Means if instead of glycine, alanine, if any other amino acid is linking, the new protein is formed. In secondary structure, it gives such polypeptide chains are exist in long form and therefore it is having two different structures, that is alpha helix and beta pleated structure. Alpha helix is just like a spring or a coil. Like that, even beta pleated, such number of spring structures are joined together to form a roof-like structure or the sheets which we can see on the top of the terrace. So like this, the secondary structure is formed. In tertiary structure, these polypeptide chains may get folded. It may get folded in a long chain or it may get folded in round shape, and based on that there are two major molecular shapes are formed, that is fibrous and globular. So it represents overall folding of polypeptide chains. Now how these polypeptide chains are held together? The main forces which stabilizes the secondary and tertiary structure of proteins are hydrogen bonds, disulfide linkages or van der Waals force or electrostatic force of attraction. Then quaternary structure. Some of the proteins are composed of two or more polypeptide chains referred as subunits. So spatial arrangement of these subunits, that is three-dimensional arrangement of subunits, is known as quaternary structure. So we can just think the example of a train. Consider train as a protein. So each bogie is considered as amino acid. If two bogies are joined to each other, the middle part where they are joined is called as a peptide bond and the two bogies are called as two amino acids, that is peptides. So by linking such number of amino acids, it forms the proteins. How the proteins are classified? On the basis of molecular shape, they are classified as fibrous proteins and globular protein. When polypeptide chains run parallel and are held together by hydrogen and disulfide bonds. Then fiber-like structure is formed. Such proteins are called as fibrous proteins. So this is we have seen in tertiary structure of proteins. These proteins are insoluble in water. Example, keratin, that is the protein which is present in hair or wool or silk or nail. That's why our hair are having a thread like structure and the hair is insoluble in water. Myosin is the protein present in muscles. Next, globular proteins. Globule means a spherical shape. When polypeptide chains coil around, then it gives a spherical shape. Such proteins are called as globular proteins. These proteins are soluble in water. Example, insulin or albumin. Now, albumin, I hope you have heard this word as egg albumin. Now what happens if when these proteins are subject to change by heating or by adding acids or by changing its pH, then its natural form or the native form get changed. That is secondary and tertiary structure, maybe alpha helix structure or maybe fibrous structure get changed, and this process is called as denaturation of protein. It is the process in which proteins lose their tertiary and secondary structure or secondary and tertiary structure by the action of heat, acids, pH, etc. It loses its biological activity also. It is called as denaturation of protein. The process is called as denaturation of protein. Example, coagulation of egg white on heating or boiling. You might have observed when the egg is boiled, the white part of the egg comes together and it forms this little bit spherical shape. This is called as coagulation of egg white. Then curding of milk, how the total milk structure get changed into curd. So these are the examples of denaturation of protein. Now here in definition you have seen that only secondary and tertiary structure is written. That is loss of secondary and tertiary structure or change in secondary and tertiary structure. But primary structure does not changes. That is, it remains intact. So the question can be asked on this part as what is denaturation of protein? Give an example. Or one more question can be asked as during denaturation of protein, which structure does not get changed or which structure remains unchanged? The answer is primary structure does not changes. So this is how in today's class we have completed about the proteins. Thank you. Please note down.